Stanford University.
Welcome to EE380 Winter 2010-2011. I'm Andy Freeman. The other organizer of this lecture series is Dennis Allison. Next week's talk is about a different way to build parsers. And parsing is always a big deal when you don't use it when you should or when you apply it correctly. So parsing is always welcome. This week's talk is about yet another way to build computational devices. However, this time it's DNA. And it's also about a way to use programming to do chemical synthesis, which seems like opposite problems, but they're not. They're sort of very similar problems. And today's speaker, Paul Rothemund, is at the forefront of addressing both. This is a big deal because Moore's law of fab costs matters a lot. And since we build computers to do real-world things, combining computation and chemical synthesis, as in actually modifying the world, enables a whole host of new applications. So basically, I guess the summary is not only is there a lot of room at the bottom, but there are a lot of opportunities at the bottom for doing things other than just move an electron a bit. We're not there today, but today's speaker is doing the building blocks that are going to get us there. Paul Rothemund.
Thank you very much. That was a really good introduction. Thank you very much. I'm really pleased to be here. And first I'd like to say, if we can, I'll take questions in the middle. Anytime anybody wants to talk about something or ask about a slide or anything like that, then make me aware and do that. So we'll keep it a little bit interactive. So as my slide talks about, I want to talk about using artificial DNA nanostructures, not natural DNA structures, for creating a new kind of fabrication. So a fabrication process that, while it will be in some ways similar to the kind of fabrication process we use to make computer chips, it will be in some ways very different. So it's not the case, although I'll talk about some examples that go in the direction of building nanoelectronics. While it's the case it may look like at times that we might be trying to compete with silicon, I think that in the end, the things that we're going to do with this kind of technology are very different. But the purpose is to imagine and envision and develop a totally different, complete fabrication process at the nanoscale. So I'm going to start talking about three different loosely associated research themes within my field, just to give you an idea of what I'm not going to talk about today and sort of how the field started. So this is Ned Seaman, and Ned Seaman started the field of structural DNA nanotechnology, which is the field of programming, and I put a little p here, structure. So and that means, this means what a sort of chemist means by programming. That means merely specifying structure, right? So he, over the course of 30 years, has developed a language for building geometric shapes out of DNA. These are models of two structures that he made out of DNA in the early 1990s. And his goals in doing this are really completely orthogonal to the ones that I'm going to talk about today. He originally wanted to make three-dimensional DNA crystals out of these kinds of building units and to put proteins in them and do a better job at electron crystallography. So now after 30 years, he's managed to get some three-dimensional DNA crystals finally and start to put some proteins in them. So Ned Seaman invented this whole field of using DNA as an engineering material from a structural point of view. So another strain, other than making structure, is putting in some dynamics. So programming function and dynamics into DNA molecules. And I'm representing this with Bernie Yerke, who's a physicist at Bell Labs for many years, another physicist at Oxford, Andrew Turberfield, and a chemist at Columbia, Milan Stojanovic. And this theme within our field goes towards putting dynamics into DNA molecules. So what you'll see today, the rest of what you'll see today, are static structures that don't move at all. They have no interesting dynamics, but it turns out that you can, just as the protein motors that move our muscles and carry bags of chemicals around in cells can walk and chemical energy can be used to make them drive interesting motion, there's a whole subfield that has to do with making DNA walkers. So this is a schematic for a cycle for how a DNA walker moves. And so there's an entire rich field of making non-equilibrium DNA structures that do interesting things, and I won't talk about that today, but there's really neat stuff there. Sort of half the field now is these non-equilibrium DNA structures. The third theme is what, is it Andy or Andrew, do you like? Hi, do you, Andy or Andrew? Andy, okay, sorry. It's what Andy alluded to, and that is this connection between computation and molecules. So I represent this with my PhD thesis advisor, Len Alleman, and Eric Winfrey, who was my postdoc advisor at Caltech. This is this idea that we could take actual mathematics problems that we're interested in and encode those mathematics problems in strings of DNA sequences in a test tube. So we might have 10 to the 15 DNA molecules in the test tube representing different parallel computations, and then do some series of biochemical steps, and at the end of the day, have an answer in that test tube to that mathematical problem. So that's doing computation with molecules. That's very different than sort of the things that I'm talking about today. Sort of the flip side of that, it's using molecules to sort of build a computer. So doing computation directly with molecules is a very different thing. And here I put a big P here for programming. This is programming in the sense that a computer scientist means. This is programming that brings in all the machinery of computer science, all the beautiful complexity theory, and in principle, and indeed in practice, you can put any computer program that you can think of into the molecules. There are encodings, and you can let the molecules run, and they will do a computation. It turns out that there was a lot of excitement in the early 90s that we might compete with silicon by making very large instances of these biochemical computations. But it turns out none of the constants have worked out right. So if you multiply everything together, sort of the cost per operation per time or whatever, this doesn't work out. So it's the case that people are not pursuing DNA computation in that same way. But it's completely morphed or transformed into something else. So what it's done is it's become something that silicon doesn't have to compete with. That is to say, a little bit of computing, just very small logic circuits, a little bit of computation allows you to make decisions in a molecular system, in a biochemical system, and control other biochemical systems. So now DNA computation and RNA computation of various sorts are viewed as a mechanism towards the control of other molecular systems, something that electronic computers don't have an interface to. And what are those molecular systems, when they do their little bit of computation, going to do? That little bit of computation is going to have them make decisions about what drug to administer. So for example, a little molecular computer is going to sense a bunch of molecules that are indicative of a disease state, do a little bit of logic that says it's this disease and not this other disease, and this is the situation, this is the drug I need to administer, and then release those molecules of interest. So that's what molecular computers seem to be good for, sort of in that sense, and that's where that third strand of our field is going. And it's also the case that there's another strand of that I'll just allude to a little bit. You can use the computation, rather than to make a decision about what other molecules should be released, you can use a computation to determine what pattern or structure can grow. And then maybe you might later use that structure or pattern for building some other device. And so that's another place that this can go. So I talked about those three strains. The sort of overall motivating philosophy behind our field is that we're going to put all these things together. We're going to put structure, as Ned Seaman has figured out, we're going to put function, things that move, or maybe even do some chemistry, and we're going to put computation together, and we're going to make things that are as complicated as living cells, right? That's the overall goal of our field. And the things that we have to replicate, the kind of things that we have to replicate, are indicated by this picture. These are all kinds of qualitatively different parts in a cell. This is a membrane. In the membrane here are things like photoreceptors and the energy-producing machinery of the cell. There's information storage in the form of DNA, but then there are these proteins that bind to it that are boxed that do the genetic logic that says what parts of the DNA program should be turned on. And then there are structural elements like those at right. These are very long, rigid protein filaments. And so these are sort of the girders of a cell that give it its form. And then there are things like this, which these are the 2 feet of a molecular motor that can walk along along these filaments. So at the molecular scale, it's got all these things. It's got structure, it's got computation in terms of the genetic program, and it's got all kinds of function. And so we're going to try to recapitulate all that. Now, historically, so I'm just going to talk about structure. Okay, so we're going to go back to structure, and historically, the first really successful artificial DNA structures that were made by Ned Seaman, and then also in collaboration with Eric Winfrey, they were used to make the first large-scale nanopatterns out of DNA. And what those structures were just two double helices. So these are sort of, looks like two double helices together. And really, these structures, we call them tiles. You can think of them as a little bathroom tile or a brick. These little bricks had four corners that had little single-stranded pieces of DNA hanging off. So there's the double-stranded part, and then there are these single-stranded parts, and these single-stranded parts had free open A's, C's, G's, and T's that could specify the interaction of these bricks with each other. So these bricks could come together, and these particular bricks, when they come together, this is just showing these are bricks that are all supposed to be the same, but the different colors show the symmetries of how these bricks come together. But the point is, these particular bricks come together at an angle. And so these bricks could form a tube that's not unlike this girder that you see in cells. So these little, this girder is made out of little protein tiles, little protein bricks. There's going to be a tube that we make out of these little DNA bricks. And so you can make different sets of these DNA bricks. For example, if you have just one DNA brick with certain DNA sequences hanging off its corners, it can make a uniform 2D lattice. If you use two kinds of bricks with different so-called sticky ends of DNA sequences, you can make a diagonal pattern, you can make a stripe pattern, you can make these things. And then when you look at them under an atomic force microscope, and I'll talk a lot more about this later, you can see that you can actually make these molecular structures in a programmatic way from these little bricks. And it turns out that so if you think about the kinds of things that you can make out of periodic structures, there's a whole, you can enumerate the kind of structures you might want to make, and that enumeration is a certain number of wallpaper patterns. There are a certain number of patterns, qualitatively different geometric ways that you can make a periodic order in 2 dimensions. And we haven't made all of them out of DNA, but for example, here's a kind of crazy one that starts with some 5-fold symmetric tiles that break their symmetry and end up with a really funny 2-dimensional fabric. It turns out that there's no obstacle to making all sort of possible 2-dimensional fabric patterns like this with DNA. So that's something we can do. And we can go further. So as Andy alluded to, this is an atomic force micrograph. So just to give you a sense of scale, this is 40 nanometers. Most of these tiles are somewhere between 10 and 20 nanometers across. Okay? So this is an atomic force micrograph where these little gray bodies are a little 10 nanometer DNA tile, and the white bodies are another little 10 nanometer DNA tile. So this is a 2-dimensional fabric made out of DNA. And it may be a little bit hard to see, but this is actually a chunk, although it has some errors in it, but it's a chunk of a fractal pattern. And that fractal pattern, the overlay doesn't work, that's okay. So that fractal pattern actually is Pascal's triangle mod 2, or otherwise known as its Sierpinski triangle. And so this pattern was grown in a layer by layer fashion. Those little DNA bricks started growing here and grew in this direction, and the set of DNA bricks that we used implemented a little binary computation and elaborated this pattern in the crystal. So it's the case that we needn't make these periodic patterns. We can pick arbitrary sets of bricks that in fact can do any computation you can think of. They can simulate cellular automata, if you're familiar with that model of computation. And so you can embed into a crystal relatively complicated patterns that you might use for something else. It's the case that this is a great efficient way with a very small number of DNA bricks to get a complex pattern. But yes?
You mentioned errors. Do you have self-error correcting triangles?
There are. So there are this particular program for making Sierpinski triangles only uses 4 DNA bricks. If you make the program larger and include some redundancy, there are techniques for making somewhat more complicated sets of bricks that include error correction that can effectively eventually exponentiate the error rate. Yeah.
Agent of the making
The agent of the making.
Yes, what is it that causes, you keep saying, we make these things, what is it that's...
So I'll talk more about that in a second. So let me remember your question and then give you how that works when I talk about the example I'm going to talk about for most of the talk. All right. So for the purposes of having a fabrication method, however, so I said you can use a relatively small number of these different chemical entities, right? A relatively small amount of chemical information to get complex patterns, but you're limited to the ones that you're limited to a certain family of patterns that are the ones that you can make by a growing that you're limited to the ones that you can get sort of using cellular automata. Okay. But if you want to have a fabrication technology where you're allowed to make an arbitrary pattern, right? You want to, if you have a layout for a chip, you want to be able to put the wire exactly where you want, right? You want to be able to make any arbitrary structure in there that you possibly can. The transistors could be anywhere, and in fact, you would like to be able to make a, sort of a random pattern. You can make anything you want. So the question is, how can we do that out of DNA? So that's the main topic of the talk, is a method for doing that. So we want to figure out how to make arbitrary shapes and patterns out of DNA. And the method that I came up with a little while ago to do that was one that I called DNA origami. So DNA origami, so before the paradigm was you have little bricks. I didn't describe very well what they are, but I'll describe these things better in a second. You have a little DNA brick. It has little sticky ends, and those ends have different kinds of glues that can stick with other bricks according to whether or not the glues match. That's a tiling paradigm. Here, this is a folding paradigm. So we start with a very long strand of DNA. So this is a long strand of DNA that we actually steal from a virus. So it's about 7,000 letters long, so it would really snake all the way around the room. So this piece of DNA is not manufactured. It comes from a biological source. We purify it from a virus. We could make it synthetically, but it's much more inexpensive and it's much more convenient to just steal it from a biological organism. Now, the goal of this technique is to figure out how to take this very long strand of DNA and fold it into whatever shape that you want, hence the word origami. And so the way that you do that is introduce a bunch of small DNAs. These small DNAs I call staples, so that immediately breaks this metaphor of paper folding, but so they're staples, and these are synthetic. And what I mean by making them, this maybe should answer your question, is there's a machine about the size of a photocopier called a DNA synthesizer, and it's got a little bottle of A, a bottle of C, G, and T on it, and you type in A, G, C, T, and it has a bunch of tubes and piping, and it washes the chemical constituents, A, C, G, and T, over a little glass column, and they bind in order, and you create the sequence of interest, and then you add another chemical, and it cleaves it off that column, and you are left with a little tube that's exactly the sequence you want. So
Effectively putting in a test tube and giving it a shape.
Okay, so that is not just putting it in a test tube and giving it a shape. That is a serial process by which we construct these small DNA sequences. So this is a synthetic sequence that's made by a company, that's a DNA synthesis house. They're sort of a foundry for biotechnology. Everybody in biology uses short DNA sequences like this. So we use them for there's an enormous industry around synthesizing arbitrary sequences of DNA. They are the input to the process that I'm going to describe today. Okay, so we live on the ability of them to synthesize these short strands of DNA for us. Okay. Short. So here I have depicted this one as being 8 long, but the ones that you'll see later, this is just a cartoon. They're all anywhere from 25 to 30 letters long. So the words are 25 to 30 letters long. Did that help at all?
I'm just wondering what brings the two things together. What brings the virus DNA and the
yeah. So what brings these together is mixing by hand. So so we grow up some virus in a bacteria and purify this long strand away for them. That's one tube. Then there are going to be 200 of these other little guys. And I sent an email to a company. I said, make me 200 of these short sequences. They come in a little box. There are 200 tubes. I mix this long strand with those 200 tubes, add a little bit of salt water, and then heat them up and then cool them down. So you anneal them. So that's the basic process. Okay. And then what makes them come together when you say what makes them come together, is a subtle question, but A's stick to T's chemically, and C's stick to G's, right? So A sticks to T, C sticks to G, G sticks to C. So for the left half of this molecule that I call a staple, it's designed by computer to bind to one part of this long strand. Okay? The right half is designed by computer to bind a different distant location on this long strand, right? So then when they do that, it enforces a constraint between these two positions on the long strand. So then the net action of 200 of these guys enforcing 200 of this kind of constraint folds this long strand into something that you want, right? That's the basic game, okay? Yes?
Big long chain that you got from a virus.
Yep.
There's nothing magic about the
There's nothing magic about it. Well, other than that, so that sequence is an evolved sequence. The virus uses that sequence for its own purposes. There are all kinds of genes in here and stuff like that. What's important is that that sequence not be too repetitive. So for example, in this case, for example, if I have this red staple and I want it to bind here and here but not somewhere else, it can't be the case that this subsequence also appears over here. So you have to get lucky and have a sequence that's not too repetitive or doesn't have too many there are other similar kinds of problems. It can't bind to itself too much. There can't be a word here that sticks to another word over here and collapses it too much. So in fact, I'll describe a little bit about that. It turns out that in this virus, there is a place that it sticks very strongly to itself, and we just don't use that part of it, right? Some people do it kind of successfully, but most of the time we just eschew that part and leave that little part hanging off unfolded. So the sequence can have problems, and in the limit that it's very cheap to make these sequences, we would like to design our own in a computer and use those, right? Because then we can optimize over this sort of subsequence problem, and we do that for those DNA tiles. They're so short, they're made out of all synthetic DNA. Those are completely computer optimized to get rid of those kinds of problems, right? All right. So for example, this thing folds under 200 constraints and makes something like a rectangle. Okay. Now this is a totally different representation. This is the black strand is that long viral strand. These colored strands are those short staples, and these little hash marks are places where an A sticks to a C or a C sticks to a G. So this schematic is meant to show all the little places where they hybridize. Okay. So I'm going to show you a cartoon now. This is a movie that is completely not representative of reality. It was made in it's a cartoon, but it's meant to give you a picture of what happens over the course of an hour and a half when we heat these things up and cool them down, right? So this is made by Sean Douglas. He's at William Shee's group in Harvard, and he very nicely made this for me. It's a movie where you have this long strand, you have a short strand, a bunch of other short strands. You heat them up, you cool them down, and so this thing will look like it has an intent, right? No movies of self-assembly that we can ever have the strands fly in and find exactly the locations that they have in a real molecule. In fact, they're binding and unbinding and binding and unbinding and whizzing around. They'll bind and another guy will push another guy off. There's all kinds of stuff happening. But this is to give you a picture of the kind of process that must happen. So in the end, you end up with a structure that has a bunch of things that look like double helices in them, all right? So it's got a bunch of parallel double helices, and it's got what we call crossovers. Crossovers are places where one of these strands winds along one helix for a ways, and then jumps across to the other helix and winds back in the opposite direction. So it's sort of like a weave out of this long strand and a bunch of short strands that enforce those constraints. Yeah?
You actually simulated this way or backwards?
Oh, this is backwards. I mean, that's what he did, right? So he constructs this thing, and then he doesn't use any kind of Brownian dynamics. He used like wind.
That's why, if you do that, that's why you get this
Absolutely.
thing.
But we can't easily do it realistically. We don't have the compute time. You can't make a movie that's the real thing. So that's why I'm trying to say this is yeah.
So you're using multiple instances of the virus DNA.
In this case, there's only one, but we will use billions. So we'll make billions of copies of this same thing.
What is the yield?
So the yield depends on many things. The yield, as far as I can tell, depends on the purity of that long strand. So in a typical experiment, where you have a structure that otherwise does not have problems, which I'll define what I mean by that in a second. If you have a structure that otherwise does not have problems, as this long strand comes from the biotech company we buy it, 95% of them are intact and pristine, and so 95% of the structures that you get are what you want. Okay? And if you did a better job purifying that starting material, you would probably do much, much better. All right, so that's my answer to that question. But the thing is that the question of yield comes up in many places here. So there are many different steps in the processes that I'll describe that each have their own yield. This step has a 95% yield so far, but I'm not worried about optimizing this in the sense that we can purify that long strand much better. There are many other problems that I think will be harder to work on. So the yield of these particular structures like this is not worrying to me, even though in the typical experiments around 95%. Okay. Okay. I mean, it is, but it's not surprising now, like 5 years later. Okay. So when I first came up with this technique, I wanted to demonstrate that, for example, that long strand wouldn't get tangled, that it could take on an arbitrary structure. So I designed. So I'm not going to show the staples. This is just the folding path of that long strand. So I wanted to have something that would take a circuitous path up and go down, reverse, go up over here, cross, come back down again, right? So it takes this long winding path through this structure and allows us to create voids, right? I wanted to be able to make things that had holes in them and show that the thing was still relatively rigid with holes. And then I told you about that piece that, who asked that question? I told you about the piece that folds on itself a little bit. You asked that question. So we leave that off on the bottom and leave it unfolded. We're going to pretend it's not there, okay? So, all right. Okay, so we're going to attempt to make this thing. So I did. I designed it and designed the staples. And just to give you an idea of the scale, right? So now we're going to zoom out. And so there's this thing. What's that thing? Okay, so that's the receding curvature of a red blood cell. Okay, so this is a 7 to 9 micron red blood cell. So about 70 of these guys fit across the width of a red blood cell, to give you an idea of scale. So they're 100 nanometers. All right. So the way we visualize these things is using atomic force microscope. We can't easily visualize them in solution, so we have to plunk them down on a surface and then use this relatively destructive technique to image them. This is a fine micromachined needle, and it's raster scanned back and forth across a surface. There's a laser beam that's bounced off this needle. You measure the perturbations of the needle as it drags back and forth across the DNA. So that's how we're going to measure these two-dimensional DNA shapes. Okay. It's a mechanical system. It's reading by Braille, sort of. You can, although we don't typically. Some very exotic AFM allows you to see individual atoms. What you see today is much grosser. STM is usually what we use to see atoms. Okay. All right. So this is again a completely fictitious simulation, but in a single drop of 100 microliter drop of fluid, there are about 100 billion of these guys floating around, okay? So to use that very invasive imaging force, dragging the tip of a microscope back and forth, we have to immobilize them. So they're in a buffer, they're in a solution that has lots of magnesium ions. Those magnesium ions both stick to the DNA, which is negatively charged. The DNA carries lots of negative charge, and also stick to the negatively charged mica surface. Mica is this mineral you find in the ground. You can cleave it and get a perfectly atomically flat surface. And so then we take an atomic force micrograph, and you'll see immediately the difference between the design smiley faces, and some of these guys have their noses punched out and their jaws flipped over their head and stuff like that. So that is, so there's a question of why is that, right? That's a component of the yield. So only 72% of these guys look like, and this, you have to figure out how you measure this, right? What do you mean by they look like the smiley face? They don't have holes that are too big, for example, above a certain size. But so these guys are not all well formed, and I'll get back to some of the reasons why that might be the case for these particular guys. But you can zoom in on one and see that he has the detailed structure that you're interested in, and he even has that little loop hanging off the bottom, right? So just for scale, here's that guy against those girders and viruses and the normal DNA, right? So you can see that. So here's a yield of some guys who are somewhat more rigid, and 95% of these guys are the triangle that you're interested in. Now, I want to remark, I guess, here, there's the self-assembly of the DNA strands with each other via the A's, T's, C's, and G's that make these shapes. But there are actually two other kinds of self-assembly that are occurring in this picture, okay? So there's the highly controlled self-assembly that makes a smiley face, but there's the self-assembly of the shape to the surface via that charge interaction with the magnesium. And then also you might look, these guys look like they make friends. They look like they kind of stick together, all right? So that's another kind of self-assembly that here you can tell is completely uncontrolled, right? So those are two other kinds of self-assembly that we can use in this system, and if I'll get to it, I'll talk about how to try to control both of those with the same sort of accuracy that we control this smiley face. Control their aggregation and control them sticking to the surface, okay? So these guys, you'll notice, don't stick to each other, all right? Yeah?
Seems like these structures sort of want to be two-dimensional. I would think that they would often the third dimensional.
So there are a couple things. You can ask, are they really two-dimensional? And so when I first made those, I designed those structures with a certain symmetry that I thought would tend to cancel out any curvature they might have. And we'll get to that. Whether or not they're 2D, we'll get to that in a little bit, okay? But that's a very interesting question. Okay. But one reason why they're 2D is they're, rock solid stuck to a surface. It's like a piece of paper glued to a surface so that tip doesn't drag it around, right? All right. So about a year after I published this method, I got this in the email. Does anybody know what this is? Somebody has to know what that is. Not you. You probably know what it is. What is it? China. It's China, right? So I'm embarrassed to say I didn't know that was China. And I got this from Lulu. Huh?
Today's groundhog.
It really does look like China, though. Here's a map of China, and this is the approximation that Lulu Shan, who is a graduate student at SJTU, made, and now she's actually a postdoc in Eric Winfrey's lab at Caltech.
Oh, wow.
So she did this. I'm sorry. She did this entirely on her own with no communication with me. She read the supplemental materials that I had. She wrote her own code to do it, and because she was sick of waiting around for me, and she just made this. And so she made this beautiful approximation of China. And here she even did something different. She still left some loose unfolded bits, and then folded a little bit that's supposed to be Taiwan, right? So that's Taiwan on the world's shortest leash, right? So, all right. So, all right. So this method, everybody always asks about 3D, for example. This method has been elaborated by William Shih at Harvard and has made beautiful three-dimensional structures this way. So this is a strain diagram, and these things fold into these 3D structures. So you can make all kinds of solid girders and cross pieces and all kinds of stuff like that. And I'll diverge into biology land for a second. So he's made some crazy sort of railroad tracks and some icosahedra and stuff like that. So we're not just interested in using this for nanofabrication for inorganic devices. William Shih is interested in, and many others, including I, are interested in making these structures as custom instruments for biology. Okay? So what do I mean by a custom instrument for biology? This thing is a little cross piece that has a little hole in it. And that little cross piece makes railroad tracks with these little holes in it. And the purpose of this thing is to bind little protein walkers. So he hasn't got there yet, but this is the direction he's going over the next years. And those protein walkers are those ones that I showed on that slide where I showed the biological molecules. They're protein motors that walk along other protein tracks. And there are many aspects of their mechanism of their action that we don't know about. So we're trying to learn a lot about them. And one of the things we don't know, for example, some people know certain aspects of this, but one thing you might not know is how it is that when the thing is walking, it knows to lift its back leg. How does it know to let go, right? So it might have a little strain sensor there, right? When it strains its back leg, it might decide to let go, right? And so one way you might access that question is you might want to make protein tracks that have different step lengths. So if he takes little short steps, then his back leg doesn't let go so often, so he walks slower. Or if you give him long steps, then maybe his back leg falls off more often and he walks faster, or he just plain falls off the track more, right? Something like that, okay? So that's what this is designed for. So yeah? You're pointing at someone. Oh, over there.
Like the group at Stanford and Carnegie Mellon who has the toolkit for building structures out of RNA, are there?
The best person in the world for making RNA the RNA structures of this kind is at UCSB. So I feel bad that I don't know who you're talking about at Stanford.
There's a game now.
Oh, yes, yeah. So that's a somewhat different problem. So here we're interested in making artificial structures to order, and they may look nothing like biological structures. In their case, they're trying to get at the question, RNA's folding is much more complicated than, it's much more complicated than DNA. And so it's much less straightforward to understand, given an RNA sequence, what it will actually fold into, which is a very important problem in biology. And that game that they're talking about is meant to get at, it's meant to give information about that process. So that's a real biological thing. But the point is that William Shih is trying to then, so it's hard to make a protein girder and change the step length. So that's a difficult engineering task. But it's easy to make a DNA girder and change the step length and ask that kind of question that I was talking about and see if you can make the thing mince its steps or stretch its steps. So it turns out that it's quite hard to get a really good foothold for the protein on the DNA. So that it actually may be difficult for other reasons, but nevertheless, this is about trying to use DNA as custom instruments for biology. Similarly, there's some Japanese guys, and I won't go into detail, but what they've done loosely is they've made a picture frame out of DNA origami, and in that picture frame, they've strung one DNA that's tight and one DNA that's loose. So they've got a tight DNA and a loose DNA, and what they're doing is they're measuring the activity of proteins that bind to these loose or tight DNAs and either cut them or don't, or mark them or don't. And so they can measure, they can now make a little instrument that's completely self-contained, sort of its own little control with a tight and a loose part, and they can show that the tight one gets cut under a certain circumstance and the loose one doesn't. Okay? So this is another custom instrument for biology that people are working on. All right, so now I'm going to go back to this patterning thing. So the general problem of making these things. So back to 3D. Hendrik Dietz, who is in William Shih's lab, has come up with a very simple model for introducing curvature and twist into these structures. All right. So by just taking a particular unit chunk of DNA, so DNA has a twist, right? So if you're building things out of, you're building stuff out of twisted plywood, right? So it has a certain angular twist per unit length. That's the equilibrium one that DNA would like to assume, all right? And if in your DNA origami structures you use a different length per unit cell in these structures, it actually exerts a little twist, a little local twist. And you can balance them to make something curved, or you can leave them unbalanced to make the thing twisted. And so he's made entire families, he's calibrated this, and he's made entire families of twisted and curved structures. So you can make really nice rings that have little teeth on them that look like a gear. They're not, yeah.
You can do this later, but these structures tend to be very stable.
Yeah.
How do these break down? Being unnatural structures, are they cleaved like normal?
No, for example, they're more resistant to enzymes that would cleave DNA. I mean, they're more resistant to enzymes that would cleave DNA, for example. But I won't get to that much later, so maybe we can talk about that afterwards. So these look like gears, they're non-functional, but the point is you can nevertheless see that he's got an enormous mastery of the ability to make whatever curve he wants, right? So he's actually calibrated this very finely, and he can dial in exactly the angle he wants in a curved sense. He can make all this stuff. So this is not functional yet, but he can nevertheless make the structures. Now getting back to your question about whether or not things are flat, so if you take my original model for how I made the DNA origami structures, and I said I thought I made them flat, right? It turns out if you use the best model that we have derived from the parameters that you extract from this kind of work, and you revisit the smiley face looks like that in solution, right? So the smiley face is in fact probably not flat, and we know that many of the other structures I've made, we can have direct evidence. You can see that they're not flat. It gives you terrible results when you try and fabricate something out of them. So now in the second generation structures, we use the model basically, it's just, we don't even use his model. We just pay a little bit more attention to the twist of these structures, and you can get them quite flat over large regions. But this may also contribute to why that smiley face looks so broken up, because when he's sticking to this very sticky surface, if part of him sticks and then the other part sticks and he folds over, it can, because his nose is so distended and his jaw is bent back, maybe that gives you some of those screwed up structures, but we're not sure. But that's at least a hypothesis you can entertain. Okay.
You were saying you just hand optimized it to be as
No. I just made a bad assumption. So we can talk about it later, but it's just a simple geometry. It's a simple geometry. We can talk about it later. Okay. Okay, so people have gone on to make three-dimensional boxes. It turns out he used the same approximation I did. His box is a little twisted and screwed up, but this is a beautiful box made by Ebba Anderson at Aarhus University. And the point of these kinds of structures is these structures have lids that can actually open and close under the influence of other molecules, and they can potentially act as drug delivery devices and stuff. So there are people who are actively pursuing that. Okay, again, not nanopatterning, but that's something that people are doing. All right. So now let's get to patterning, right? So how do you make a pattern with these shapes? So here's the long strand, here's the staples. There's a one-to-one correspondence between each staple sequence and where it ends up in the final structure. So this green strand ends up exactly there, right? And this orange strand ends up exactly there. So if you want to put a pattern on top of this, you want to encode a pattern on top of this, then on the staples that you're interested in, you put some kind of biochemical mark, and then they end up exactly where they're supposed to in the final structure, right? So by marking some subset of these strands, then you can get a pattern. And so these are atomic force micrographs of some patterns that I made on DNA rectangles. So these are 100 nanometer rectangles, and the resolution of these patterns, the pixels in these patterns, are about 5 or 6 nanometers, right? So you can approximate some snowflakes, you can write things, and getting back to your question of yield, actually there's the yield of the overall structure, but here you can see the D is missing a pixel. And some of the other ones are missing pixels. So that's another yield you have to worry about. In this case, the yield of this, in terms of detecting those modifications, is about 95% itself, right? But we're not sure because actually when you're doing atomic force microscopy, you can watch these things blink out and get rubbed off. So it's not clear, it might be better than that, but certainly that's a lower bound on how well you're doing. So the yield comes in a lot of different places. Okay. So you can make patterns at the nanoscale. It seems like artwork, but what's artwork really for? Well, making artwork allows us to make computer chips, right? Computer chips that are essentially printed. And so the fantasy is this, right? The fantasy is you put some components on the DNA staples, you heat the thing up, and then you get some little circuit. And then you asked about DNA stability. Well, if you're doing subsequent processing, once this is down on a surface, you don't care if the DNA gets pyrolyzed and burned away. Maybe we wash the DNA away and you're left with some kind of functional device, right? So that's the fantasy. So there are lots of problems with that. This is a chart that I don't expect you to understand at all, but it's from the semiconductor industry has a big group that they pay a bunch of university PIs to explore alternative fabrication technologies. So that's called FINA, and they drew this graph here. In this column is is the normal lithography. There are a bunch of alternative things based on protein, based on bacteriophage or DNA like I do, and block copolymers and other stuff. And the point is, in red and in yellow, right, are supposed to be potential showstoppers for using this thing for any kind of nanofabrication, right? So if you zoom in, so this is my column using DNA to do this. It's got some red and yellow in it. And what are the problems? There are things like device compatibility, yield, like you're talking about, defect rates, trying to scale up to larger structures. The structures I showed you so far only have 200 pixels in them. What can you do with 200 pixels of complexity, right? And registering them with other things. How do you possibly register these with other kinds of fabrication, right? So I'm going to try and get to talking about how these things are maybe a little bit white and not such a problem, or maybe red is yellow. So we're going to try and work on those problems. Okay. All right, so in terms of active devices, getting active devices on these things, it's of interest to organize carbon nanotubes with these, right? So can you make a carbon nanotube device with these? Now that's been done before, sort of in 1D. You make a 1D device, you can combine DNA with carbon nanotubes, and you can use the surface as a backgate, and so you can make devices that way. But the real question, which was done in this collaboration at Caltech, really driven by these students, is to organize carbon nanotubes in 2D, right? You don't want to just use a single long strand of DNA. You really want to be able to make some kind of cross. And so the picture then is you have a DNA origami rectangle that has one kind of DNA strand on the top and one kind on the bottom. And you make two kinds of carbon nanotubes, right? So the blue ones and the red ones. And the blue ones have one kind of little DNA widget on them that's not exactly just the complementary DNA strand. And the red ones have a different one on. They're these protecting groups. But they're released if and only if these carbon nanotubes bind. And the point is, you want to get crossed carbon nanotubes. Now, if you start with a mixed population of semiconducting and conducting carbon nanotubes, and you're lucky enough to get a conducting one and a semiconducting one, then you can make an FET with this. right? And you could purify the populations of carbon nanotubes to try and make just the red ones just semiconducting and the blue ones just conducting, but we didn't do that for the purposes of this experiment, right? So then you can make some DNA origami, and here you can see this DNA origami actually has some other stuff attached to it. I won't talk about that, but this gray DNA origami has some crossed carbon nanotubes on it. And you can see there's no control over translation because they're crossed near their ends, right? So that's another problem of the technique. But nevertheless, you can get some, you can take images of this and you can wire them up. So these are gold electrodes wired up. There's a little DNA origami, or maybe the DNA origami in this case we think may have been removed in one of the processing steps. You have two carbon nanotubes that are crossed, and that cross was made by associating them on a DNA origami, and you can use one of these carbon nanotubes as the gate and the other one as the channel, and you can demonstrate switching behavior. Okay? All right. So that's great. Now, the students worked very hard and they did an awesome job with this. And then you ask, how does this compare to taking a bunch of carbon nanotubes and throwing them on the ground and finding crosses, which is what everybody did in the late 90s and early 2000s anyway. And the fact of the matter is you made these things in solution. So they're floating around in the tube like those smiley faces, and they go all over the ground, so you have to find them anyway, right? So you have to go find the crosses that are made with the DNA. And in solution, it's the case that the DNA origami can bind to more than one carbon nanotube. Right? So then they can cross-link the structure. So if you try to push the chemistry to get a higher yield, then you end up getting networks of devices, and they're not in some nice rectilinear pattern that you might like, right? So then you have to go and find the devices and write, electrodes to these things to find them, right? So that's no fun, right? So this is this problem. This is this problem of registration. The smiley faces and everything goes all over the place, right? So how do you address that? So in a collaboration with IBM Research Almaden, which is down the road, we took a very simple approach to doing that. So we tried to place triangles where we wanted them. So they have a substrate layer, some kind of template layer, and a photoresist, and then just use E-beam or optical patterning to pattern the template layer. And by patterning the templating layer, I mean simply using an oxygen plasma etch to burn away the template layer, right? To the substrate below. And so what you're left with is a substrate with some background in yellow and then spots that are the shape of DNA origami written by E-beam, okay? And these spots, in the presence, so they're supposed to be binding sites for DNA origami. So in the presence of enough magnesium, we have the same kind of charge effect we have on mica. So the spots are nominally negatively charged, the magnesium is positively charged, and it cements the DNA origami on those spots, right? That's the goal. And we can do this with just chemical differentiation between a silicon dioxide surface and a trimethylsilyl monolayer, or in diamond-like carbon, there's a little bit of topography and also a chemical differentiation, right? And so I won't go into the details of this too much, but the point is that you can put DNA origami where you want them on a surface, right? So here we have alternating columns where they were supposed to be pointing up and where they were supposed to be pointing down, the triangles. And you can go ahead and you can measure their orientations, and you can see how well you do. And on the diamond-like carbon surface, for example, 95% of the sites, again, that's just fortuitous, as far as we could tell, had single origami, and their standard deviation of their angle was, they were oriented within plus or minus is supposed to be 10 degrees, right? So it's actually fairly broad distributions. Here's a column that are all supposed to be pointing down. There are many issues with these very first experiments we did. So one of them is in the case where we had the best data, the size of the triangle is a little bit, the size of the sticky patch for the triangle is a little bit too small. So the triangle is a little bit too big for the sticky patch, so you can imagine the orientation wouldn't be perfect just on those grounds. And if you make a movie, here's an AFM movie of different sized triangles, different sized sticky patches binding DNA origami as they come on and off the surface, and then they get ripped out by the AFM tip. And if you look at this movie, the triangles that match their cognate sticky patches have exactly the right size, they appear to stick longer and have better orientation, obviously, than if you have things that are too small, where they don't stick very well, or things that are too large, where a bunch of them stick, right? So all this hasn't been completely worked out because we're actually in the throes of trying to reproduce a lot of this at Caltech in my lab right now. So anyway, so they do better when they match. All right.
So some years ago, we had some folks from Alien Technology. They commented the water
laboratories doing the same thing.
Is this the big roll-to-roll thing where they have all the little...
No, this is making very small pieces of silicon, breaking them off, and then putting them in, making a template that they drop in. They use a water slurry to put it in.
Right, I mean, they have all these little pieces, and you see the thing going by, and they're like little raindrops, and they sit down into their...
Fluid flow.
Yeah, it's fluidic self-assembly. So as far as I know, I think just gravity and just a cognate fit drives that.
And
Fluid tension.
Yeah.
Surface tension. In that case, since they're in a bulk fluid, I think surface tension doesn't come into play in that particular one. But that's so how are we going to put these things together? So we have this problem of aggregation of the DNA templates by components, and you don't know where they are. So the kind of thing that we're trying to do now is make shapes that are DNA origami that are asymmetrically shaped, put them, make cognate sticky patches on the surface, put them down first, and then assemble the devices on top of them. So if you put the carbon nanotubes on after you've put the DNA structures down first, then the DNA structures can't be aggregated by the devices, and you also know where they are. So you can use very regular kind of lithography, very sort of easy, cheap, much more cheap lithography to lay down a grid of electrodes or whatever else you might want to do. So that's the sort of long-term goal for that. And so the other aspect of that I should point out is that those triangles that worked so well the first time, they have too many symmetries. So those triangles can rotate in 3 directions, and they can flip over, right? So it's the case that you don't want your devices to go down in all these different orientations. So we're going to try to use highly asymmetric shapes to get devices down in exactly one orientation. So my goal with all of this is to eventually be able to give somebody a wafer, and that wafer has a bunch of these sticky patches on it at different distances and in different configurations, and then hand them a bunch of DNA origami and say, well, your job is to connect your device of interest, whether or not it's a carbon nanotube or a piece of graphene or a quantum dot or something like that. Your job is to connect that to the DNA. And then if you wash these, well, your job is to get that chemistry to work out. Wash the DNA over the surface, the DNA origami, go where you want, and then place your devices exactly where you want to have many instances of some little device that you want to study, okay? And so here, I think it's not the case, so biology actually can benefit a lot from that, but also for physical scientists, it's not the case that we're necessarily going to compete with silicon here, but if you want to study some materials that are not normally in the silicon process, you want to study some colloidal semiconductor quantum dots or carbon nanotubes or things that don't necessarily mix very well, or some titanium dioxide particles because they do some neat photochemistry. If you want to make a little device like that, you can potentially use this technology, right? You can mix a lot of different materials as long as they can all be coupled to DNA. So you're not competing with silicon, you're trying to make it a lot easier for you to study the devices you want to make. So that's where I want to go with it. Now, we're just trying to place some asymmetric DNA shapes. So these are some funny asymmetric L's. Here's a really nice rigid right triangle we made, and we made a bigger floppy right triangle that's not working so well. But so we're doing that, and we're trying to reproduce that placement technology right now, and so I can talk about that offline. People have placed things other than carbon nanotubes. So Jen Cha, who's now at UCSD with Albert Hung, have placed little gold nanoparticles, right? So here are the DNA origami triangles with little gold nanoparticles, and they've placed them at their tips on a surface on one of these grids using this placement technology. Now, they put the gold particles on the DNA origami in solution. And so some of the aggregation you see here is due to the aggregation in solution. And you can also see places where 2 origami are actually stacked up on top of each other. So in their hands, they found that it wasn't that good in their situation to put the particles on afterwards. But other people with other particles have succeeded in doing that. So this goes in that direction of placing different materials where you want. And the kind of thing that you want to do there is make optical devices, right? So I think one of the places that will prove this kind of technology first is in making optical nanostructures. And that's because, for example, unlike for carbon nanotubes or nanorods, we don't have to solve translational symmetry problems. We don't have to get them to line up in translation on our structures where we want. We can rely on metal nanoparticles going down at individual points, right? So it's the case, so here are spectra, either scattering spectra or I think these are just scattering spectra simulated scattering spectra of a single 50 nanometer gold particle ring of gold, 6 gold nanoparticles, and a ring with a gold particle in the middle. Now, these kinds of structures have been studied before, most recently by Fan et al. In Science. They were using more fancier particles, but the point is that the way people normally try to make optical devices out of these structures is they throw down a bunch of particles and wait and hope to get lucky to find them in this configuration and then study this guy, right? So the point of this technology is to use the DNA to organize it into a whole bunch of guys that all have the same configuration of gold nanoparticles. And the neat thing about this particular series of things is that, for example, just single particles have a high frequency resonance. Hexagons of particles have a single, much longer wavelength resonance. But if you make a little resonator of metal nanoparticles like this out of silver, it's hard to see. There's one peak here superimposed with the red one and another peak here. And then between these peaks, if you put an array of these on a surface, as we hope to do with placement, you should get negative index of refraction material. So for a specific range of frequencies, we should be able to construct a negative index of refraction material in the visible by using DNA to place these little clusters of metal nanoparticles. So that's sort of, I hope to get here in the next couple years, but this is a direction we're going. And another interesting device direction, I think, is if we take one of those triangles that I showed you, like one of these asymmetric right triangles like this, we hope to put little metal dipoles on these, wherein these little metal dipoles, so this little pair of particles has a very different resonance than a single particle, and its resonance is polarization dependent. So what I've tried to draw down here is that there's a resonant mode that's parallel to the long axis of the particles and one that's perpendicular to it. So if you excite with light that's polarized in this direction, then you have to excite with red light to get good scattering, or in this direction with blue light. So, or if you polarize the light in this direction, then if we had white light polarized in this direction, then we would see these guys as red and these guys as blue. So that is to say, we can change the polarization, and based on the orientation that we've placed these metal dipoles using DNA structures, we should be able to get different colors. And that's been done before with lithography. All right, so I should just say, people have placed little metal dipoles of little metal disks, and based on the orientation of the little metal disk, they can get a different scattering spectrum out based on the polarization. So that's all been done by lithography in the early 2000s. But we want to recapitulate that with this directed self-assembly, because with one pass of E-beam, we can lay out a field that will allow us to place these particles, right, and get one set of optical nanodevices. But then to get a different kind of optical nanodevice, all we have to do is change the DNA strands. So the difference between, this is a triangle with 2 metal particles, and this is a triangle with 3 metal particles. The difference between these two is just changing 3 DNA strands and mixing them up. So you don't have to go back to lithography to change the device that you want to make, right? You change the DNA and you change the device that gets laid out on the surface. So in this way, we hope to make a standard lithographic substrate and then be able to make a whole variety of devices based on the DNA organization. So we have our own. Yeah, and it's terrible. It's not showing up. So there's this green line, just for fun. This is kind of a joke, right? So if you count the number of distinct DNA bases in the structures that we're making and take that as a measure of the complexity of the thing, then that follows, that's an exponent, and the doubling time is, I think, supposed to be a couple years. I don't know why this line it's not on my computer either, but it's the case that, at about 2008 or whatever, this is the 15,000 nucleotides that represents the DNA origami that we've made, and out here would be a couple times that. It's the case that we've already made things with about 5 times the complexity of the DNA structures that I've showed you, right? So we have 10 minutes left, and there are 2 choices. Either, sorry. Right, no, yeah. So this is a slide. No, yeah, right. So the things that are off the line are qualitatively different experiments that don't have to do with just making shapes, right? So these are things that walk and move and stuff like that. Okay, so we have a choice. You have 10 minutes, and either I can talk about one way of increasing the number of pixels, increasing the number of complexity in a pattern that we have, or we can just have questions. What do people think? Okay, all right.
We can have questions afterwards. There's no further.
We have room until tomorrow morning at 8 a.m.
I read in the thing, it said we had to run out the door at
The camera stopped.
The camera.
Oh, okay, all right.
Camera stops and then you can tell us the truth.
And then we make a video.
All right, so one of the problems with this technology, as I told you, with this particular way of making DNA structures, you get 200 pixels at 6 nanometer resolution in a little 100 by 100 playing field, and then that's it, right? So how do you compose those into more complex things? There are many ways to do that. One involves just putting DNA strands that are complementary to each other on the edge of these things and clicking them together like tiles. Now, people have done that, and that's interesting, but for other reasons that I hope to get to, there are other ways to do it that are kind of illuminating about the basic nature of the DNA binding interaction, okay? So this is all done by my graduate student, Sung-Luk Woo, and he just submitted it, and he's very happy, so this is really good. So here's a DNA rectangle, here's a smiley face, here's a triangle, and if you look at them, the rectangles aggregate into these long chains, the smiley faces aggregate into these somewhat hoary-looking ugly chains, and the triangles are all loose and dispersed, right? And you can ask, what's the difference between those, right? So here they are sticking to each other. And the difference is, if you look at the way that, so DNA is a double helix, but you can also at the nanoscale think of it as a rather stiff cylinder. So it's a somewhat stiff cylinder. And in these rectangles, the cylinders all run parallel like this. And in the smiley faces, the cylinders all run parallel like this. But in the triangle, the cylinders are running in 3 different directions, and none of their ends are actually exposed. So all the ends of the DNA helices are hidden in this triangle, right? So the ends are what are sticking to each other, all right? So the flush cut ends of the cylinder, which don't have these A's, T's, C's, and G's, right? So normally the 2 strands come together, the A's meets the T's and the C's match the D's, okay. So this is a DNA helix, and you can ask the question, what makes DNA stick together? What makes 2 DNA strands stick together? And what is anybody's answer to that? Anybody have an answer to what makes 2 DNA strands stick together? That's all right. So
Depends on the open end.
Huh?
Depends on the open
Well, no, but that's a subtlety. You already know that's subtlety. So that's good. So the thing that you read that everybody universally answers is hydrogen bond. So here's a double helix. There's this strand. This is the chemical group. This represents either A or G. I can't tell, and this is either T or C. I can't tell from this angle. But there are hydrogen bonds between the A, C's, G's, and T's. You know, you may have read about the hydrogen bonds that hold it together. So there are these interactions across the strands that hold the thing together. But it turns out that if you measure it, the greatest contribution to binding energy of 2 helices is actually a lateral interaction in this gap between 2 pairs of bases. So this pair of bases is sticking together and fit together. This pair of bases is sticking together and fitting together. But actually there's a very strong lateral interaction between this pair and this pair as a pair, right? So it's like...
They're not parallel.
They're very close to parallel.
Well,
I mean, well...
at least it's said they're not.
Well, I mean... So I mean, they're different things. So, I mean, in DNA, they're relatively close to parallel, and they're at a very low angle with respect to this. They're almost perpendicular. In RNA, they're shifted a lot. I mean, it depends on the details of the...
Questions that induces some long-range properties. Anyway.
Okay. So, but let's entertain the possibility. There's one point of view that says what makes the strength of the interaction, not the specificity, but the binding strength of the interaction of the 2 strands coming together is this lateral interaction between this base pair and this base pair, right? So in fact, depending upon how you calculate it, based on different experiments, it could be 80 to more than 100% of the energy of the strands coming together, right? So the bases confer specificity and say which strand gets to stick to whom, but what's providing the strength is this stacking interaction. So that means if you have the edge of a DNA origami and the edge of another DNA origami, there's the end of one cylinder here, the end of another cylinder here, the ends of the cylinders stick together, all right? So if you have a bunch of cylinders in parallel, the ends of all those cylinders stick to each other like this, right? So they all kind of meet. So that's what's making these origami aggregate. So that's trying to diagram that here. So here's 2 of these cylinders, and actually the ends are sticking together. Now, if you think about it, if you make some rectangles, and they're made of all these cylinders, and you just let them stick together, they could stick together in principle in all these different orientations. So that's what I've diagrammed here. They can be flipped, they can have some kind of translation, right? So they can come in 4 possible different orientations, and they can do this. So the question is how to break that symmetry and turn this into a specific bond so we can make what we want. And so what my graduate student came up with, we came up with 2 ways. One way is this. So these are 3 origami. Along the edge of the origami, you either put in a DNA staple that makes the ends take on a rigid geometry and are 2 little cylinders sticking out, or you leave a staple out and it leaves that scaffold an unstructured little loop that balls itself up, like so you leave this little unstructured ball here. So now you have a sort of a binary sequence of either structured bits that can stick to each other or unstructured bits that should have, in principle, not a very strong interaction. They can have some interaction. And then you put binary codes of those along the origami. And so here are 5 different origami that have been combined, and the yield of this is the yield of these is like 30% or something, right? So here yield comes in again. It's the yield of how well they combine together, right? So you can make binary codes, and here again, if you want to come up with good binary codes so that this guy sticks to this guy but not this guy, right? Then you need a computer program to do that. And in general, constructing such codes is formally NP complete, but what? Sorry? No, it's formally NP complete, but you can get a small set of good codes fairly easily, and so that's what he did. Especially since we're only putting 5 together. But if we wanted to put hundreds together, it would be much harder to get good codes. But he's come up with good codes. There are here you can make them form long chains where they're all oriented correctly, and they do have errors. And the kind of errors that they have, unfortunately, are mostly these off-model interactions. So here we constructed a code with the following property. The code has a property that if the binary sequence of these edges lines up, it either matches at all 7 places. So it either matches at all 7 places where these little rigid cylinders, right? And that's strong enough at this temperature for them to stick together. Or for any possible translation, it only matches at 4, right? So, and 4 is too weak under these conditions for these things to hold together real well. But the kind of off-model thing that happens is these cylinders are not perfectly rigid. So your binary code can bend. So here's 5 guys binding, holding these guys together, and you can see these little ones here have bent to match, right? So the off-model, so you can try and put that into the sequence finding thing too to try to find ones that can't bend, but right, so that's the kind of thing we can do. And the other way that he's done work to do this, other than these binary codes, is a shape code. So here he has a shape code, which doesn't show up very well. These things are jigsaw puzzle pieces. And these jigsaw puzzle pieces have one of 3 depths. So there are 4 patches along their edge with 3 depths. It's like a Manhattan skyline stuck on its side. So it's a Manhattan skyline, and the Manhattan skylines fit together. And you come up with codes by a similar method, and you can string together 4 different DNA origami that have complementary binding interactions so that you can't even see the suture line between these jigsaw puzzle pieces when they come together. But again, they suffer from bending problems. So these were done so that either they matched at all 4 patches, or they could only match at 2, right? And 2 is too weak and 4 is strong enough. But here they match at 3 because one of the patches is bending. So this is, again, is this sort of off-model thing, and the yield of this was, I think, like 45% or something like that. So anyway, that's the end. NSF Computer Science has been very good to us. The Semiconductor Research Corporation through the FINA Center has been very good to us. Microsoft Research gave me some nice funds to start with, and Mark Sims gave me some funds to buy DNA. And so that's it. Thank you very much. What do we think?
Oh, I have a question.
Yep.
Does this sort of thing show up in biology?
By sort of thing, you mean?
The same sort of structural replication and construction seems to me to, it's surprising that it hasn't evolved somewhere.
So, So I showed 2 different paradigms for making structures out of DNA today. One was this tile-based paradigm where we have a unit that tiles space and sticks together and kind of crystallizes. The other one is a folding paradigm. That crystalline paradigm occurs very often in biology. Like those protein girders you saw, viral shells are like that, are crystalline. This particular, as far as folding is concerned, each little protein in biology, or each little functional RNA in certain things like the ribosome, is actually folded. Okay, and they self-fold. You have a long sequence of information, chemical information, that folds on itself to make a structure. This method, where we have a long strand and a bunch of small ones, is actually doesn't appear to occur anywhere in nature that we know of, and it's conceptually distinct because in the first one, with the way proteins fold on themselves, all the information is contained in that individual strand. In the second one that we're talking about today, the information is contained not in a long strand. If I just hand you that long strand, you don't know anything about what you're going to get. You need to have the joint staples and that long strand. And so that paradigm of folding things does not appear to appear in biology.
Can you guess what kind of impact it may have on real biology if they encounter your machination?
They eat it. I mean, you mean what happens if we put this thing in real biology?
Yeah.
The greatest impediment to do anything useful with this stuff is keeping biology from eating it, basically. I mean, that's, if you want to know what... No, I mean, they just... No, they just have enzymes to chew. I mean, look, if you take bacteria... If you take bacteria and put them under starvation conditions, they'll eat DNA. They can use that as a carbon and energy source, and they chop it up. These are just things made out of DNA, and their enzymes may be somewhat less efficient at destroying these because of the compact structure, but nevertheless, given enough time, they get destroyed or hydrolyze themselves, and then they're just food.
Well, let's just put this way. You mentioned the machine that you type in some sequence
Yeah.
and puts the powder.
Yeah.
The rumor was that some mad employee typed a few numbers in, made the stuff, put it in his boss's coffee. The boss got sick. Anyway, that's the story. I'm not sure.
I mean, that's definitely a sign. It's a beautiful William Gibson story. There's a beautiful William Gibson.
Like somebody maybe, but who knows?
Yeah.
I saw that bacteriophage was on that semiconductor chart. Are there parallels between what you're doing and the bacteriophage guys are doing at different scales, or is it entirely different?
It's totally different. So what they're doing is they're engineering the proteins to bind inorganic components like carbon nanotubes, or so proteins have much more richer and interesting chemistry than DNA. Okay, so DNA can catalyze chemical reactions. DNA can do interesting chemistry, as can its cousin RNA. But protein, on the face of it, you can do much more interesting chemistry. And in particular, a lot of the people like Angela Belcher, who are combining viral proteins with nanoelectronics, is they're using the protein to nucleate the growth of inorganic materials. So specific semiconductors or specific materials for their batteries that those proteins can nucleate, that would be we may not even be able to construct a DNA that can do that kind of thing. But a funny feature is the following. So if you take Angela Belcher, she uses exactly the same virus that I use. She uses M13, right? And it's had many lives in biotechnology, but we use the DNA inside to make these DNA origami. Then they use the proteins on the outside to nucleate these different beautiful inorganic materials. So my joke about that is, between the two of us, we use the whole buffalo, right? We use the whole buffalo, the inside, the outside, all the parts, right? A dumbbell.
You can make discs, you can make rods, and you now have a way to make things. So, I did only go this way. If you had a rod and a
So
discs.
I have not made dumbbells. So
Because after you make a dumbbell, you can make a log cabin.
yeah. So such a thing has been kind of made. This is not by me. Here, no, here, no, here. This thing, that's a dumbbell. That's a hole. It self-assembles where this dumbbell rod actually is interdigitated with that hole.
But that's a sort of different construction technique of starting with discs and rods. And you can start with discs and rods.
Yeah, but I mean, he could have. I mean,
Yeah.
this is the same technique, he just chose to do it that way. That is to say, you can pick many, given this, it's sort of like clay, you can pick many different architectures and work with them. If you suspect, for example, that for the length scale you want to work at, they're going to be more rigid than something else we've done, then you're totally free to do that. And that's a good kind of way to go.
So you pick this particular chunk of virus to start with?
Yeah.
What happens if you use something else? Is there anything interesting about it?
Yeah, it doesn't necessarily work so well. So we got lucky. So William Shih, who does this 3D work, has put enormous effort. So there's another natural sequence, another natural virus you can use, Phi-X. We've used that in the lab. It sticks to itself a lot. It doesn't make origami quite as well. Then William Shih has gone and played the game and said, well, this natural sequence, it's no good. I'm going to make a much better artificial sequence, right, using my cleverness with designing the words that are not redundant and don't stick to themselves. And so far, the artificial sequences that he's made have not worked as well. And it doesn't mean that in principle there's any problem. It may just be that...
In evolution, it's of years to play your programmer if you let your
Was this
programmer...
your merit, though, for evolution?
Yeah, that's the real play.
I think what it means is that particular virus happens to have sequence has certain statistical properties, like the fraction of A's versus CG's, that sets the melting temperature of the total structure in a nice place for the protocols we're using. In principle, what you want to do is you do want to design these sequences as computers, and if we get the statistics of the sequences right, set the melting temperature right and everything, there won't be anything special about this sequence.
I can imagine that if the DNA strand sticks to itself too much, it would interfere with its function. It might be inherent in useful DNA that it doesn't stick to badly.
It absolutely is, and you can see that in the evolution of different bacteria that are meant to live at 120 C versus room temperature. You can see that in the evolution of viruses that are meant to be whose genomes are single-stranded versus double-stranded, where they don't even have the problem of sticking them to each other. And so the statistical sequence properties of all these different things across different bacteria and different viruses capture evolution has many different reasons for skewing these statistics over the first order and higher order statistics for single nucleotides, double nucleotides, triple nucleotides, for all those different species. And so maybe if we got lucky with M13, it's not the case. It's the case that there exists many other viruses and bacteria that have sequence statistics that are totally different than M13 for their own special reasons. So we might have got lucky with M13 for this thing, we might have, but it's not necessarily general, because this other virus has its genome with it's sticking to itself for certain biological reasons and has different statistics, and it's perfectly evolved for its thing, right? Yeah.
Since you're talking programming, it's known that you can execute DNA both forward and backward. Apparently, viruses are more likely to take advantage of them. Did you come across that part? Does it impact on your
So our virus
instruction?
is a relatively, so there are viruses, and I can't remember the one, there's a virus that not only does it,
higher code density.
yeah, it is. So there exists a virus that has a sequence that's read in the forward direction, but not only is it, and in the backward direction, but not only is it read in both directions, but for example, words in DNA, in the genetic code, come in 3-letter words, right? That's read in 3-letter words. And normally for a particular sequence, it's only read in one so-called frame. So the genetic program, the genes are encoded just in one set of 3. In one virus, it reads in the forward direction. The backwards direction of the program also makes sense, but also all 3 reference frames encode different proteins, as if, you took... Yes, right? It's like, your program in your computer memory, you shift by 1 bit and it's still a program and it does something else, right? That's crazy.
They can read 6 different ways.
Yeah. So our virus, the one we're using, is not that compressed. It's not that compressed.
But it may have an impact on the behavior. Certainly some of the statistical entropy, whatever, pick any information theoretic measure that may or may not have an impact on your...
I am shocked that virus that has that I'm shocked that exists. And so I actually would like to look at that and see what the biology is.
I'm not shocked. I'm looking for those, but anyway. For that kind of reason, that's when I heard the code density, I went, my God. There must have been some information theoretic reason that this high code density is useful. Normally, would say it's like
merely having a program genome.
You don't want to be too dense, otherwise it's most likely to screw up. But apparently the virus has long enough time to figure out that there is advantage, and that's part of the question.
So I guess what I was trying to get at with the other answer was that viruses and bacteria different ones are as different as giraffes and whales. And so, for example, some viruses are so structured, or bacteria are so structured, such that they have very high mutation rates. And it's one of the things that's responsible for why AIDS is so hard to deal with. Others have relatively low mutation rates. Some would rather replicate faster, or some it's okay to replicate very slowly. So for example, it's always, if you want to replicate faster, you'd like to have shorter DNA sequence. You want the program to be shorter. So if you compress all your genes in this funny way that we talked about, then you get to have a shorter genome, replicates faster, and if you're going to occupy that niche, it's okay. But you may suffer a higher error rate. So you may not have the error tolerance you would have as if you expanded your genome. And because viruses are different as giraffes and whales, different viruses are selected for different things. You know, compact genome, high error rate, not very good error correction, maybe better error correction, but slower replication or something. I mean, that's really, it's really amazing, but that's really the case.
Hierarchical some of what you just said may be based on different error correcting architectures, if you wish.
Yeah, there are certainly at least a couple we know about. Certainly at least a couple levels.
Human system is a deep hierarchy. Very low level code corrections, and there's some very high level.
Okay.
It's good.
People have gone away and now we can do the cool stuff. So I just want your microphone.
Oh, good.
We don't have to leave.
All right.
For more, please visit us at stanford.edu.